Good afternoon, everyone. Welcome to the first Ethel Browne Harvey Postdoctoral Seminar of the new season. My name is Martha Echeverria Andino, and I am the new postdoc trainee representative on the Society for Developmental Biology Board of Directors. We have an exciting lineup of speakers for the 2023-2024 season, and we hope that you will join us throughout the year. Now I'm gonna turn it on to Stanley Michi Kanai, which is our first moderator for today.
Thank you, Martha, and welcome again to the Ethel Browne Harvey Postdoctoral Seminar. My name is Michi Kanai, and I'm a postdoc at the University of Colorado Anschutz Medical Campus. I will be moderating today with Elisa Zhang, a postdoc at Stanford University. We are excited to highlight the work of our outstanding postdoctoral members, and today, Casey Griffin from New York University and Alain Garcia de las Bayonas from the University of California, Berkeley will share their research. Each speaker will give a 20-minute talk, followed by 10 minutes of Q&A. If you have any questions during the talk, please enter them in the Zoom Q&A box. And now, I'm excited to introduce the very first speaker of the season, Casey Griffin. Casey received her PhD from the University of Southern California, where she studied the development of parasites and their role in maintaining the blood-brain barrier in neurodegenerative diseases. She is now a postdoc in the laboratory of Jean-Pierre Saint-Genet at New York University, where she is investigating how a spliceosome factor controls neural crest biology and craniofacial development. And for this work, Casey is supported by an NRSA F32 fellowship from the National Institute of Dental and Craniofacial Research. So please join me in welcoming Casey Griffin. Casey, take it away.
Thank you, Michi, so much for that introduction, and thank you to the organizers for giving me this opportunity to share our work with all of you here listening. My project and today's talk is entitled The Function of SF3B4 in Neural Crest and Craniofacial Development. In the Saint-Genet laboratory, we study neural crest development in the context of numerous craniofacial disorders. And the craniofacial disorder I am interested in is Naegger syndrome. Naegger syndrome is a form of acrofacial dysostosis, which includes midface protrusion, micrognathia, absent thumbs, and radiohypoplasia. This is a rare disorder whose frequency is unknown, but there are around 100 documented cases of Naegger syndrome. Because of the effects on the craniofacial region in these patients, specifically the midface protrusion and the micrognathia, affecting the neural crest-derived structures, this disorder is classified as a neurocrestopathy. The neural crest are a highly migratory transient cell population that delaminate from the neural tube and migrate along the body axis along distinct streams to populate various regions of the body. They give rise to numerous cell types depending on their position along the body axis. And in the craniofacial region, these cells give rise to, amongst other things, the bones of the craniofacial skeleton. So in Naegger syndrome, the regions that are most affected are the neural crests that populate pharyngeal arches 1 and 2 that give rise to the craniofacial structure. Now, for majority of the patients who have Naegger syndrome, they have been shown to have mutations in a gene called SF3B4. SF3B4 is a member of the spliceosome. The spliceosome is a ubiquitously expressed protein and RNA complex that functions in all the cells of our body to identify pre-mRNA, find the introns, splice out these introns, and join the exons to form mRNA. SF3B4 is part of the U2 subunit, which functions in 3' intron recognition and recruitment of the catalytic subunits. Within the U2 complex, SF3B4 interacts with other protein binding partners, structural RNA, as well as the target mRNA. Now, the big question we have is, how does a mutation in a splicing factor that is ubiquitously expressed lead to a neural crest specific disorder? And to answer this question, we have a hypothesis that SF3B4 has neural crest specific targets and or binding partners, and upon mutation, these interactions are disrupted or lost, which leads to the Naegger syndrome associated craniofacial defects. Now, to study this hypothesis, we have chosen to model Naegger syndrome in two ways in the lab, both in vivo and in vitro. So I will go through both of our work in both of these systems. I'll start with in vivo. We use the model system Xenopus tropicalis, which is closely related to the more well-known Xenopus laevis. When we first looked at endogenous expression of SF3B4 in Xenopus tropicalis, we saw that at stage 17, which is pre-migratory neural crest stage, the expression of the neural crest is shown here in SOX10 staining, and we saw that SF3B4 is enriched in a region encompassing the neural crest region, but goes beyond that specific region. When we looked at the migratory neural crest stage, later stage 25, we saw that SF3B4 was enriched in the branchial arches and craniofacial region. So to further study SF3B4 in Xenopus tropicalis, we decided to make a CRISPR-Cas9 mutant line. So here we're showing in blue the coding sequence of SF3B4 protein, which includes in red the RNA recognition motif domain. And in yellow, we show our guide RNA, which targets the beginning of the protein right at the first start codon. This results in a 31 base pair deletion that ultimately leads to a frame shift and an early stop codon. The resulting protein product goes from a protein with a 389 amino acid sequence to a protein with a 29 amino acid sequence that is of the complete wrong sequence. We show here by Western blot that our mutant embryos have lost expression of SF3B4, while the heterozygous maintain SF3B4 expression, although it's probably less than the wild type. We just haven't fully quantified that. Interestingly, we also saw that there's a head size difference across the genotypes. Looking at the tailbud stage, we see that the average head length and head width are both affected in these embryos, in the nulls and in the hets. So to begin our studies, we wanted to see what is happening with the neural crest. So we looked at the neuralist stage, which again is the pre-migratory neural crest stage. And we interestingly saw no reduction in neural crest markers. We're showing two here, SOX10 and TF-AP2 epsilon, and there's no change in expression intensity or expression domain in any of these markers across the genotypes. However, when we look at tailbud stage, which is the migratory neural crest stage, we start to see some changes. We see that the neural crest stream length is diminished in both the hets and the nulls, and the presence of the streams, the intensity of the staining is also diminished. So we are losing some streams, and the streams that we do have, some of them are reduced in length. This is quantified here for SOX10 expression, and we see that while this effect is variable across the streams, we do see statistically significant reduction in some of the streams across the markers that we've tested. We also saw at this stage that there was an increase in apoptosis with the loss of SF3B4. So in the head region, again, in our wild types, we saw no tunnel positive cells, but when we look at the hets, we start to see an increase in tunnel positive cells and a larger increase when we look at the nulls, showing that with the loss of SF3B4, we see an increase in cell death in the head region. So we wanted to see next what's going on at a more global picture in these embryos. So we decided to characterize the global impact of SF3B4 depletion on pre-mRNA splicing. So to do this, we did RNA sequencing on whole embryos at stages 15, 25, and 35, which represent pre-migratory, migratory, and post-migratory neural crest stages across the 3 genotypes. And then we can do analysis of the RNA-seq data to determine not only differential gene expression, but also changes in mRNA splicing events. So the events we are interested in are skipped exons, retained introns, and alternative 5' or 3' start sites. And what we saw was that overwhelmingly the splicing events that are impacted are the skipped exon events. This is showing, these graphs are showing at stage 15, the null versus the wild type and the null versus the heterozygous. And the skipped exons are the overwhelmingly the most affected and most changed in between the genotypes. And this is a pattern that continues on through stage 25 and again into stage 35. So we've started to mine these data sets to look for terms that are of interest to us and genes of interest to us. And so when we looked at the differentially expressed genes, for instance, here at stage 35, the downregulated differentially expressed genes, we pulled out terms of interest such as a decrease in neural crest migration and a decrease in negative regulation of apoptosis. So these terms we've pulled out coincide with the data we've seen within the embryos ourselves. And so this is the type of analysis we are looking to continue with as we move on with this project. So now I'm going to take a turn and go over to our in vitro modeling of Naegger syndrome, where we use human embryonic stem cells to model neural crest differentiation in a dish. So we take embryonic stem cells and push these cells to become neural crest cells. So our starting cells are ESCs shown here with E-cadherin positive cells and SOX2 positive cells. And we can push these cells to become neural crests, which express AP2 alpha and P75. This is a schematic of our differentiation protocol, where we take the ES cells, put them into the differentiation medium, and they develop into neural ectodermal spheres. These spheres begin to settle a few days later, and then they will give rise to migrating neural crest cells that come out of the spheres. And by day 12, you can get a pure population of these neural crest cells in the dish. To study the role of SF3B4 during neural crest differentiation, we use knockdown via siRNA, where we target the cells during early differentiation, adding siRNA day 2, day 3, and day 4. And this results in the knockdown of SF3B4, as shown in these Western blots. On day 4, we see a reduction of SF3B4 expression. And on day 12, we see a complete loss of SF3B4 expression in our system. So what we saw was in our control, with a control siRNA, we see the same as in untreated conditions, where we get nice neural crest forming. We get neural ectodermal spheres that settle onto the dish and give rise to these migrating cells. And these spheres look very round and healthy before they give rise to the neural crest. But when we knock down SF3B4, we start to see some changes. Here we're showing these big lawns of dying cells, where the majority of the cells that come off of the neural ectodermal spheres just die. And here's a picture of a neural ectodermal sphere that is actually giving rise to dying cells, which you can see as these tiny blebs along the edge of the sphere. We were able to collect a few cells that weren't dead from this conditions. And what we saw was via Western blot, a reduction in P75 and HNK1, which are two neural crest genes. So we're seeing a reduction in the neural crest formed, but we saw an increase in P53, which is a sign of increased cell death. When we carried out this differentiation a bit longer, this was all done at day 7. When we carry it out a bit longer to day 12, the picture is still very much the same. Our controls show nice lawns of migrating neural crests coming out of the spheres, as shown in these first two figures. And again, the spheres are very nice and healthy. But when we knock down SF3B4, again, we mostly see dead and dying cells coming out of the spheres, but the few cells that do survive take on a neuronal phenotype, as shown here, which is precocious neuronal differentiation in this system. And the spheres, again, look very unhealthy, giving rise to dead and dying cells. So, in summary, today I've shown you 2 different models of Naegger syndrome, in vivo and in vitro. In the in vivo system, we've shown that a loss of SF3B4 results in no loss of neural crest markers at the neuralis stage, but results in reduced neural crest streams at the tailbud stage. And a loss of SF3B4 results in increased apoptosis in the head region at tailbud stage. Our loss of SF3B4 also results in changes in RNA splicing events, which have pulled out such terms as a decrease in neural crest migration and an increase in apoptosis. And for our in vitro system, we've shown that a reduction of SF3B4 results in aberrant neural crest formation with reduced number of neural crests formed, an increase in cell death, and a possible bias towards neuronal fate. So for the future directions for this project, for the in vivo side, we are looking to analyze the mutant SF3B4 line at later stages to examine the craniofacial cartilage development, as well as analyze the candidate genes we get from the RNA-seq data. And for the in vitro, we are looking to examine the differentiation potential of neural crest that have loss of SF3B4, as well as use our in vitro neural crest system to examine binding partners of SF3B4. And we hope that the combination of the in vivo and in vitro models will help to sort of pinpoint the underlying mechanism of the pathology of Naegger syndrome. So with that, I would like to thank the members of my lab, especially my PI, Jean-Pierre, for all of his help and guidance. I'd like to thank my funding and all of our collaborators. And I, again, would like to thank the organizers for allowing me to present my work today. And thank you all for listening.
Thank you, Casey, for that wonderful presentation. We will now begin the Q&A session, and I want to remind you all to, if you have any questions, please enter them in the Zoom Q&A box. Okay, so we have a question from right off the bat. So this is from an anonymous person from the audience, and they ask, how early does the migration defect appear in your null mutants? And does the loss of SF3B4 impact the EMT process?
So we are not quite sure how early the migration defect starts right now. We saw it at stage 25, which is the tailbud stage. And we are currently waiting. We receive embryos from our collaborators because we don't house the frogs in ourselves. So we're waiting to receive embryos from stage 20, which is we're hoping to be able to gain a better picture of when this migration defect happens. And as far as EMT goes, we're not sure if this affects EMT. We are looking through our data sets, our RNA sequencing data sets right now for some EMT markers. We haven't seen anything that's jumped out yet at us, but it's definitely something we're keeping in mind as we go through the data.
Okay, and we have a couple more questions to get through here. So we have another question from Thomas Foreman, and he asks, what do you think explains the increased differential splicing changes at stage 25? He thinks it was that stage.
Oh, because it goes from like a couple hundred to like 1,000. I know what you're getting at. So I think part of that has to do with just an increase in sort of the amount of differentiation that's happening at that time point. New systems are developing. This is a time point in development where is a big jump in a lot of the differentiation of the body system. And so I think there's just a lot more genes being expressed compared to stage 15. So that could be why there's just this large jump in the amount of genes that are affected, because there's just a large jump in the amount of genes that are being expressed.
All right. So the question box is exploding here, so we're gonna keep going. Okay. Anonymous attendee asks, are the cells that make up your neural ectoderm spheroids heterogeneous?
They are slightly heterogeneous. They kind of they're akin to the neural plate. So they have distinction. If you think of this sphere, the center as being the center of the neural plate, and as you move outward on this of the sphere, there's some regionality to the cells. If you were to take the spheres and chop them up, you'd see regional identity of those cells, and that's why the edge cells are able to give rise to these neural crests. So if you keep the spheres long enough in culture, the center cells are giving rise to neurons because they're neuronal.
All right. And here's one from Andrew Lopez. Have you checked whether SF3B4 is also misregulated in human samples?
Like human, I guess human patient samples. We haven't gotten human patient samples. That's definitely something we want to get. We're working right now to try to see if we can get a human IPS line of Naegger syndrome, because that would ideally be great for our studies. But yeah, there's been work on patients. About 60% of the patients with Naegger syndrome have SF3B4 mutations and haploinsufficiency of SF3B4.
All right. Here's one from, apologies if I get this incorrect, Shenbei Chang. Since SF3B4 is expressed in the neural tissues of Xenopus, do you see defects in neural development, as in maybe neuronal differentiation?
So in our in vitro system, we see precocious neuronal differentiation. The cells that do survive and migrate out as neural crest-like cells do give rise to neurons. And we've also, in our RNA-seq data, pulled out neuronal differentiation terms. It seems like neuronal differentiation is increased when you lose SF3B4 expression. So there is some effect with the neural system going on there. Yeah.
Okay. Somia Rao says, great work. I am also working on SF3B2 and see similar results. And they're requesting to, can you show the slide with streak that is defective in hets and nulls? And what happens to hets since there is haploinsufficiency?
I think it's this one. And so the hets are a bit variable. There are some hets that when we analyze them look a lot like wild type, and then there are some that look a lot like the mutants. The patients are haploinsufficient, so it's interesting that the hets don't always have the mutant phenotype, but there's more, it's more than 50% of the hets have a mutant phenotype. So it seems like something's going on in the hets. But yeah, we're still trying to pump up our numbers a bit to fully analyze these embryos. Yeah.
Okay, and here's one from Tim Cox. How well do you feel the embryonic stem cell experiments mirror the disease process? In embryonic stem cells, you remove all SF3B4, whereas in disease situations, there's SF3B4 levels are simply reduced. So how do
yeah, so we're using an siRNA, which is not a complete knockout. It's a knockdown and it's mosaic. So don't expect that we're losing all of our expression. What I think happens is that the cells, there's so much cell death that is taking over the situation, and I think it's more of a compounded effect, and we're not seeing it's kind of like an exaggeration of what is happening, which is why we would like to get an IPS model so that we can look more closely to the disease state by using a patient cell. But yeah, we know we're not perfectly recapitulating the disease state with the siRNA, but yeah, it's kind of an exaggerated version.
Okay, this one is from Ann Corsi. Interesting work. What types of mutations have been identified in Naegger syndrome patients? And I guess what other types of mutations? And are there any missense mutations? And are they all predicted to be null alleles?
So there are the mutations that are known for SF3B4 kind of run the gamut. There's missense, nonsense, frame shifts, it kind of all over the place and all along the protein. It's sort of all of it results in haploinsufficiency of the protein. As for the other patients that don't have these mutations, I don't know if they're known. They're not published that I know of. But yeah, that's a majority of the patients have these SF3B4 mutations. But yeah, they kind of all run the gamut, and pretty much all of them result in kind of just a loss of the half the protein, haploinsufficiency. So yeah.
Okay, and we have another question from Tim Cox. We're gonna abbreviate this a little bit. And he asks, it's a question regarding the impact on of the loss of SF3B4 on splicing. Does your exon skipping category, does it include full exon skipping, or do you also see some partial exon skipping as well?
So I believe it's just full exon skipping. Partial exon skipping actually falls into the 3' or 5' adjusted sites. I believe that's the way it goes. We have a collaborator who does the actual bioinformatics analysis, so I'm not 100% certain, but I believe that it is full exon skips. And so to answer your question, we do probably see partial skips. They just fall into the other categories.
Okay, well, thank you everyone for all of your questions. I think we've run out of time, so I'm going to hand this over now to our second moderator, Elisa Zhang. Thank you, Casey.
Thank you.
Hi, everyone. Thank you, Michi, and thank you, Casey, for a wonderful talk. Thank you to the attendees for some really excellent questions. It is my pleasure now to introduce our next speaker, Alain Garcia de las Bayonas. He is currently a postdoc in Nicole King's group at the University of California, Berkeley. Alain is currently supported by a prestigious long-term fellowship from the Human Frontier Science Program. Prior to joining the King Lab, Alain completed his bachelor's, master's, and PhD from the at the Aix-Marseille University in France, where he received the highest honors for his work. He completed his dissertation in the group of Thomas Lequi, looking at spatiotemporal and quantitative control of Rho1 activity by GPCR signaling during tissue morphogenesis in Drosophila. And today, Alain will be telling us about his work looking at other GPCRs in the closest relatives of animals. So thank you again, everyone, and we're looking forward to your talk, Alain.
Thank you so much, Elisa, for this kind introduction, and thank you everybody to be here today. I would like also to thank the organizer for allowing me to present this work. So I'm going to tell you a bit more about what I've been doing during my postdoc, in particular the functional characterization of a cell membrane receptor in an organism called choanoflagellates, and the fact that this receptor might be important to control their sexuality. So to start with, I would like to present you a bit broadly what our main interest in the lab, which is actually to understand how animals originated. And the approach we are taking is a comparative approach. In fact, we are studying the closest relatives of animals called choanoflagellates that you can see actually as a sister group of animals here on this phylogenetic tree. And this comparative approach is very important for us because by comparing the biology of choanoflagellate with the one of animals, we can infer on the biology of the last unicellular common ancestor of choanoflagellate and animals. And from there, I'll try to reconstruct the evolutionary path leading to emergence of animals. So now I'm going to tell you a bit more about these enigmatic organisms that are choanoflagellate. So in this video, you're going to see actually one of these choanoflagellate cells. So what you can appreciate is that they are single cell organism, aquatic organism. And they present on the top of having a cell body, they also have a collar that surrounds a single flagellum. And both actually the collar and the flagellum are really important for the cell, for the motility of the cell in the water, as well as to attract bacteria. So these organisms are feeding on bacteria, and the collar and the flagellum are essential for that. So it might look like a very simple organism like this, but in fact, they are able to show an extraordinary developmental plasticity. And that's something that is well exemplified by the model choanoflagellate we are studying in the lab called Salpingoica rosetta. So if you look at them in culture in the lab, they are going to appear like single cell organisms. However, they can actually transition under certain conditions to form this multicellular structure called rosette, beautiful structure where all the cells are clonal and attached to each other. And this is actually triggered by some environmental bacteria in their environment. On another aspect, if you constrain these cells mechanically, they can actually retract their flagellum and retract their collar and transition into what looks like now almost like an amoeba. And this cell can now crawl onto the surfaces. And this is actually a phenomenon that is reversible. Another environmental cues involving either starvation or some bacterial enzyme secreted in the environment can also act as trigger to push the cells towards sexual reproduction. And during these developmental transitions, the cells are going to change the morphology, fuse, and eventually produce a zygote. So all of this, I think, is showing how important the environment is to control actually the development of S. rosetta. However, there is one aspect that we still don't know about is how at first these cells manage to sense these environmental cues in order to go towards one developmental response. And so when I started my postdoc, I became increasingly interested in trying to characterize receptors that could allow these cells actually to sense these various environmental cues in order to choose a developmental fate. And so what I decided to do was to look at one of the biggest families of receptors, which is actually called the J protein coupled receptors, that we knew previously were found in quanoflagellate, but never been functionally characterized. And so I'm going to tell you more now about one of them that we name Cupidon, which is actually the French for Cupid in English, and you will understand why soon. So what you can see here is that this receptor shows the hallmark of J protein coupled receptor, so in the sense that it exhibits 7 transmembrane domains. But you can also appreciate that its N-terminal tail shows some domains that are either involved in cell-cell or cell matrix adhesion. And for this reason, this receptor belongs to a subfamily of GPCR called adhesion GPCR. Now, the reason why we decided to look at this GPCR in particular is because we realized that this receptor is conserved in at least 9 quanoflagellate species that we know of. You see here a part of the phylogeny of quanoflagellate, and at least 9 species show a conservation of this receptor. And for this reason, we believe, well, if it's conserved, it might be important for the biology of quanoflagellate. So what's its function? So we were very lucky because this receptor is conserved in our model quanoflagellate, S. rosetta. And this is great because we can now do CRISPR-Cas9 gene editing, and so we can actually assess the function of the genes. So what I ended up doing was actually to produce 2 truncated variants of this receptor. One that conserves part of a protein domain here, and another one cleaner here that actually lost actually all of the protein domain. And the first thing I wanted to know is whether actually upon the loss of this receptor, we can see some defect in terms of cell growth, because this is going to inform us whether the cell, for example, can go entirely through their cell cycle, as well as if they can feed correctly onto bacteria. And here is the result of this analysis. So you see the cell density over time. And essentially what you see is that there is no difference between the control and the mutant lines, suggesting that this receptor has nothing to do with some control of cell growth. So next, what we thought to investigate is whether this receptor could be actually involved in this transition from uni to multicellularity that I mentioned previously. So some bacterial molecules can actually push the cells to form this beautiful multicellular structure called rosette, but are they still able to do so if we now get rid of this receptor? So here I'm showing you actually control Y-type cells without bacterial inducing signal, or with these bacterial molecules. And what you can see, I hope in this picture, is that they transition from being single cell to forming this actually beautiful multicellular structure. That's actually something we can quantify here. Now, what's happening in the background of the mutants? Well, they still actually are able to form this rosette despite the absence of the GPCR. That's what you can see here. So with the induction, with these bacterial molecules, they still form rosette, and that's what we quantify here too. So something you cannot appreciate here, however, is that I have been sticking the cells onto a substrate to be able to image them. But it became extremely clear that if you let them freely swimming around in their culture, soon enough, a phenotype is going to emerge specifically in the mutant culture. So what you're going to see here on the left panel is a Y-type culture. And here in the middle and on the right panel are actually the 2 mutant lines. I'll let you appreciate what's going to happen over a period of 30 minutes. What you're going to see here is that in a space of 10 to 30 minutes, the mutant lines are going to start forming a clump. So the cell quickly aggregate with each other, which is something that is not observed in the control line. So this we can quantify over time. So this is exactly the same time lapse in the control and the 2 mutant line. And so we can quantify the area of clump, and you see that 10 minutes are enough for the clump to emerge already. So this was actually quite intriguing for us, because if we think about it, we did the knockout of a gene that presumably is involved in promoting cell adhesion because it has this conserved domain that are known to be important for cell-cell or cell matrix adhesion. And yet, if we get rid of this receptor, the cell gain an ability to attach to each other. So how does these things work? And we were, of course, very interested in what will be the biological implication of this receptor. What's the biological function associated with this receptor? And I must say we were, I think, very lucky because in fact, it was not the first time that aggregative behavior has been reported in this chronoflagellate species. In fact, previously in the lab, 2 other studies have shown that S. rosetta can also form aggregate under various conditions. One of them being when exposed to a bacterial enzyme called eros, where the cell quickly form clump. The other condition, environmental condition, is when the cells actually start starving and they also form clump. Now, despite the different environmental cues controlling aggregation in these 2 cases, it's extremely interesting to see that, in fact, these 2 different phenomena are actually part of the same biological process, which is actually the entry into the sexual cycle of S. rosetta. So here I might explain a bit that under happy conditions, when the cells have a lot of food, they reproduce exclusively asexually, so they are all clonal. However, under condition where the cells are either exposed to this bacterial enzyme or under starvation, they start clumping and eventually follow 2 different developmental paths that eventually result in the obtention of this zygote here. And so in both of these situations, the cells fuse, merge their DNA, recombine, etc. So because clumping behavior is observed in our mutant, we wonder whether, similarly to these 2 different studies that has been shown previously, whether the mutant could somehow relate to a similar process. Is this could be the sign of an entry into the sexual cycle. So we decided to look at further into the structure of this clump by comparing actually how the cells attach to each other in our mutant, GPCR mutant, and how does this compare in the 2 situations where the cells can form clump either in starved wild type or in contact with this bacterial enzyme. And so what we decided to do is actually to look at the nature of this contact between cells in a clump by classifying the cell contact into 3 categories. Either the cell contact by the cell body, or the cell contact by the collar, or the cell have a collar cell body contact. And so the result of this analysis is the following. What we can see is that in fact, the GPCR mutant cells, similar to star wild type cells, seem to adhere mainly either collar cell body or collar, suggesting that the collar is actually the sticky interface. And this is in stark contrast with the 3rd situation where the cells are actually induced by this bacterial enzyme, where this time the cells seem to attach mainly by the cell body. So this first experiment suggested that perhaps the GPCR mutant kind of, mirror a bit what might happen under starvation in a wild type context. So to assess whether the collar indeed was the sticky interface important for the cell to attach to each other in both situations, GPCR mutant and starved wild type cell, we decided to remove the collar and see whether the cell was still able to clump. So how did we do that? Well, we treated the cell with a drug called latranculin A. So what latranculin A is doing is actually to depolymerize actin, and the macrovilli of the collar are actually actin enriched structure. So if we depolymerize actin, we remove the macrovilli, and so we lost the collar. And that's what you see here in this video. In a control, you can see the cell swimming around, you can see the collar and the bacteria attached onto the collar. Now, if we treat the cell with latranculin A, we lost the bacteria attached onto the collar, and that's because the collar is actually strongly reduced. You can also notice, nonetheless, that this does not affect the flagellar beating of the cell. So, does the starved wild type cell still able to clump if we get rid of the collar? The answer here is no. You see that, in fact, we are losing the clump in this situation. That's what we quantify here. On the opposite, for the cells that were induced by eros, we say that they seem to contact each other mainly by the cell body. If we get rid of the collar, the cell still form clump, and in fact seem even to form bigger clump. Now, if we ask the same question in the context of our GPCR mutant, it becomes very clear that if we get rid of the collar, the cells are not able to form clump anymore, similar to what's happening in the starved wild type cells. So, since it seems that the loss of the GPCR seemed to modulate the adhesive property of a collar, we wonder whether we could actually have an idea of where the receptor localizes in these cells. So what I did next was actually to do an endogenous tagging of a protein. So what I did was actually to add at the C-terminal end of the protein a little tag that secondarily is going to be recognized by a nanobody. And this allows us to look at the localization of this GPCR in our cell. And what we observe is that, in fact, this GPCR localized at the collar. So what I told you since now is that, in fact, we uncover a receptor that is collar specific, and its presence allows the cell to not attach to each other. And upon the loss of this collar receptor, now the cell attached specifically to their collars. And this is actually similar to what's observed at the entry of the sexual cycle under a condition of starvation. So to go further, of course, we wanted to know whether the cells were not only able to clump, like in the starved white type cells, but if they were equally able to fuse after that. So that's the assay I decided to do next. So the assay is the following. What I did was to co-incubate two populations of cells, cell expressing green fluorescence, cell expressing pink fluorescence, co-incubate them at a 1-to-1 ratio in either a white type background or in a mutant background where the cells are clumping. And what I did was actually to incubate this cell for a certain period of time, and then look specifically for this kind of event where the cells where some cells are showing actually the two signals. So you see here a pink nuclei together with a green cytoplasm. Because these cells, if they exhibit the two signals, are necessarily the product of cell fusion. And so then we can actually quantify the extent of cell fusion in these two backgrounds, wild type and mutant. Now, the result of this analysis is the following, that in fact, we observe a 5-fold increase in cell fusion in the mutant background compared to the wild type. So this tells us that not only the cell aggregate with each other, but they fuse with each other, similar to what's observed in a starved white type condition. Now, this is not enough, of course, to claim that sex is happening. What we would really like to know is that now, okay, the cells fuse with each other, but do the nuclei are still able to fuse with each other, and can we see actually some meiotic recombination happening there? So I'm afraid that today I will not be able to give you the answer because that's something that is ongoing, but hopefully soon we will be able to answer this. Instead, what I want to show you now is that actually, so we not only look at the similarities between our GPCR mutant and the starved white type condition at the cellular level, but we also try to ask whether at the gene expression level, we could also find some kind of similarity between our GPCR mutant and the starved white type cells. So I perform an RNA-seq analysis on this mutant cell, and the result is this. So here is a volcano plot. On the right panel, the genes that are upregulated in the mutant. On the left panel, the genes that are downregulated. And you can see that actually I color-coded some of these genes, and this color actually indicate whether the genes are either upregulated or downregulated in the starved white type cells. So in green are the genes that are upregulated in starved white type cell, and in pink, red, sorry, or orange, I cannot really see very well, but in orange, the cells that are, the genes that are downregulated in starved white type cells. And so it was really striking to us to see that, in fact, the genes that are upregulated in the mutant GPCR are also the genes that are upregulated in starved white type cells. And reciprocally, the genes that are downregulated in the mutant cells are also the genes that are downregulated in the starved white type cell, suggesting that not only at the cellular level, but also at the gene level, there is a similarity between these two situations. So I would like now to summarize all what I've been telling you by showing you a schematic. So today I've been introducing this GPCR that we call Cupidon, that we've shown is localized at the collar in S. rosetta. And what Cupidon is doing, we believe, is actually in condition of high nutrient, is repressing sex. And it's doing so by repressing some sex gene, inhibiting the ability of a cell to attach to each other with a collar-collar interface, and inhibiting the ability to fuse. And in that case, the cells only reproduce asexually. Now, in the condition where we get rid of this receptor, what we have done with this mutant, we relieve actually this repression. And as a consequence, sex genes start being expressed, the cell attached to each other, fuse with each other, and now we believe that the cells are engaged into sexuality. And this shows a striking parallel with what's observed in Y-type cells under starved condition, where equally the same set of genes are going to be expressed. The cells are going to attach into the collar and fuse with each other, resulting in sex. So we don't know yet whether in a Y-type cells under low nutrient conditions, the receptor might be, downregulated, or whether the ligand perhaps is affected, but that's actually some of the research we are doing now. To put this back into a broader context, particularly in the evolution of mating, it's interesting to see that most of the eukaryotic organisms actually are able to undergo sex. But for most of them, they favor asexual reproduction and only under certain conditions shift to sexual reproduction. And so that means that this is an active process that is induced under very defined environmental conditions. But what we are showing here is that in fact in Choanoflagellate, or at least in this species, sex is actually the by default mode, and it needs to be repressed constantly if we want the cell to reproduce asexually. And so this receptor actually, when the signal is turned off, is now allowing the cell to reproduce sexually. So it's a repression rather than an activation. So that's interesting to think about. And I would like to finish.
Alain, you have 1 minute.
Perfect. So
you.
the last thing I wanted to tell you is that actually we are characterizing now the downstream signaling event happening downstream of this receptor. In particular, I mentioned that we've done some RNA-seq analysis. So we know the nature of the genes that are differentially regulated in this background, as well as in the star Y-type cell. And we are very excited because we find among these genes some that are actually broadly conserved among the eukaryote, including some gamete fusogen molecule, some transcription factors that could be important for the transition to haploid to diploid, as well as some retrotransposons that are silenced during the sexual cycle. So we believe here this study could help us understanding perhaps the evolution of the germline in animals and trying to understand actually the genetic basis of it. With that, I would like to finish by thanking really the whole group. For sure, my postdoc mentor, Nicole King, but really all my colleagues for their support during the course of this work, and as well as my funding agency. Thank you again for listening to me, and I'm very happy to answer your question if you have some. Thank you.
Thank you so much, Alain. That was a really fantastic talk.
Thank you.
As people are typing their questions, if you don't mind, I'd like to start with something from the beginning of your talk. So of the two mutants that you showed, one of them, I noticed that the point at which it was knocked out was sort of in the middle of that laminin G domain. And I was curious to know if that was performed for biological reasons or for technical reasons. And if it was performed for biological reasons, what's so special about sort of the middle of that laminin G domain and what people understand in terms of its contributions to this function. Thanks.
Yeah, so it's mainly for technical reason because we are using CRISPR-Cas9 and we are limited by the where the PAM sequence are along the sequence. So at first I went for this PAM sequence for other reason, but of course it was still not very clean because there is always a possibility that this remaining protein domain might still have an effect. So to make sure that is probably not the case, we produce this more truncated version, this time that excludes most of the domain. And it reassured us because we observe the same phenotype. And so, yeah, it's mostly technical, but the function of this domain is unknown in the case of this protein so far. But they all relate to cell-cell or cell matrix adhesion. In fact, laminin, like EGF domain, are known to bind to glycoproteins. And this perhaps can give us a hint of what could be the ligand of this receptor. But for now, we have no idea. Yeah.
Great, thank you so much. We have some questions coming in. So our first question is from Jessica Feldman. She asks, any idea how cupidon gets downregulated in the collar in response to low nutrients? So, and as sort of a secondary question to that, have you verified that it actually does get downregulated in the context of low nutrients?
Right, so that's something, so we don't know yet. And that's something that we're investigating at the moment. So until recently, we didn't have this tag protein, but now we do. And so this will allow us to assess either by Western blot or directly visualizing it, to see whether we can see a difference at the level of a receptor in a context of starvation. So it could be that the receptor is either not expressed anymore or it's mislocalized, but it could also be that the receptor is still present and instead the ligand might be missing. So that's a piece of evidence that we are lacking at the moment, but we're working on it and we're really hoping that we will know more about it soon.
Great. And Jessica has a second question, which is, how conserved is this protein? Is it broadly used or selected on across evolution for mating? And she would also like to comment that she really likes the name.
Oh, thank you. So, no, that's a great question. So it looks like this receptor is lineage specific. So we find it only in chronoflagellate. So, but having said that, when we look at the genes that seem to be differentially regulated downstream of a receptor, these genes are actually conserved across eukaryotes. And this is actually something that now that we are increasingly interested in the evolution of sex, very often the trigger of a pathway is actually something that's going to be extremely specific to the lineage looking at. But the outcome of it, the core regulatory pathway, so what led the cell to fuse with each other, as well as what's going to help the recombination event, is actually amazingly conserved across eukaryotes. And so the whole challenge for those who are interested in the evolution of sex is trying to actually narrow down what will be the core of this process that is shared by many. And so sure, this receptor doesn't seem to be shared, but the downstream regulators seem to be. Yeah.
Great, thank you so much. We have another question from Andrew Lopez. He says, great talk, really interesting story. I was wondering if you had any thoughts on why plated samples still aggregate even with Cupidon mutation.
Sorry, do you mind repeating? I'm not sure I understood.
Oh, no problem. So Andrew was wondering if you had any thoughts on why plated samples are still aggregating even with mutation of Cupidon.
A plated sample, you mean the when I grow them into... Oh, sorry. Oh, okay. I think I understand now. So why do they adhere when... Yeah. So how does it work, right? Because this is supposedly an adhesion molecule. We get rid of it and they stick to them even more. So let me show you. So at the moment, it's just a hypothesis we have, and I'm glad that you asked it. So two models we have, and they are not exclusive, and it could be something else. But we believe that either this receptor is actually masking an adhesion molecule at the membrane. And so when we get rid of this receptor, we uncover this adhesive molecule that now do the job of attaching to neighboring cells. This will be hypothesis one, the model you see here. Another possibility is that, in fact, upon activation by the ligand, this receptor is going to, perhaps downregulate the expression of some adhesion molecule, or perhaps inhibit the post-translation modification, inhibit the activity of this receptor at the membrane via post-translational modification. So these are two models we can think of, and until now we haven't been able to discriminate between both, but that's also something we are working on, and we would love to know more about it. It does seem that it's not as direct as uncovering an adhesive molecule. It does seem that actually it involves a level of signaling, and we actually more and more getting interested in cyclic GMP signaling, that seem to underlie the phenomenon. So it could be indirect and possibly modulating actually another adhesive molecule that we don't know about for now. Yeah.
Great, thank you. I think we have time for one last question from an anonymous attendee who's asking, do the mutants have a disadvantage to find food sources on the other side of the plate compared to the wild type?
So they do not seem to have, so they seem to feed properly, because what I've been showing previously, the growth curves of the mutant compared with the control, we do not see actually any change in the growth rate of a mutant culture by comparison with the control, which is something that we will expect to see if actually there is a feeding issue where the cells, clearly we can see it in a lab, if we do not spike enough bacteria, the cells are taking longer to reach to plateau. So it doesn't seem to be the case. So make us confident that probably this is not happening. There doesn't seem to be a feeding defect in this mutant. Yeah.
Great. Well, thank you so much, Alain and Casey,
you.
for your excellent talks. The seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, October 13th, when Yuchuan Miao from Harvard Brigham and Women's and Carrie Price from Yale will be presenting. And thank you all for coming today.